All right, everybody. This lecture is the second lecture in this, for chapter 6. And what we're going to do here is sort of briefly discuss genetic mapping and what that means. And then we're going to run through a linkage and mapping problem to show you guys how you solve those things. So, genetic mapping is something that's not really done anymore, but we're going to talk about it anyway, because it's important to understand how it's done to sort of understand how genomes are organized. So genetic mapping is essentially, or was, I guess, essentially a technique whereby scientists figured out the order of genes along chromosomes. And you can do that because each gene has a specific place or location known as its locus on a chromosome. So this is an example of a highly simplified map of the Drosophila genome. There's only 4 chromosomes in fruit flies. And if you look at these chromosomes, so this is chromosome 1, also called the X. These black lines along this chromosome are meant to represent different genes. Flies are diploid, just like human beings. So each fly, for example, if we look at chromosome number 2 here, each fly would contain 2 versions of chromosome number 2, and this gene, Dumpy, will be at the same place, position 13, on both of those chromosomes. And that's what a genetic linkage map is. It's just assigning positions of genes on chromosomes. Why isn't this done anymore? Well, essentially because now, for about the last 10 years or so, really, we've had the ability to relatively easily sequence entire genomes. And if you know the entire DNA sequence of this chromosome, then you know where all the genes are, very specifically to the base pair resolution. So we don't really do genetic mapping anymore, but it used to be done a lot. And it's important to understand what it means, which is basically figuring out where these genes lie along these chromosomes, or these linkage groups. So it allows us to, genetic mapping allows us to figure out not just the order of the genes, but how close they are together. Because if you think about recombination, right, you're swapping bits of DNA from one homologous chromosome to the next one. So the closer 2 genes are together, the more likely they are to move together. If genes are a big distance apart, they're more likely to get split up by a crossing over event or recombination event. So genes that are close together are often inherited together, and therefore close genes result in very few recombinant offspring, whereas genes that are far apart, there's just more probability they'll be separated by a crossover event. And therefore, genes that are far apart tend to be present at higher numbers in recombinant offspring. Now, you can actually work out what is called the recombination frequency. It's also called the map distance. Those are equivalent terms, using fairly simple numbers, in that you just do a whole bunch of crosses, you count up the number of recombinant offspring, and just work out the percentage of them versus the total offspring. So you take the number of recombinant offspring. As I said earlier, that's always a lower number than non-recombinant offspring. And you divide recombinant by total, multiplied by 100 to give you a percentage, and that gives you the recombination frequency or map distance between 2 genes. And we'll look at how you calculate that in more detail in just a second. The units of recombination frequency or map distance are called map units, MU, or centimorgans. Thomas Hunt Morgan is the name of sort of the godfather of Drosophila genetics. And so centimorgans is the name that some people give to map units. same thing. One map unit is equal to the equivalent of 1% recombination frequency. So there's several different terms there, but they all, there's a lot of redundancy with those terms. Now, I've talked a little bit about recombinant offspring and how that number is always lower than non-recombinant offspring. And some important facts to remember as we go forward are that if a pair of genes has a recombination frequency of less than 50%, they are linked. If they have a recombination frequency of greater than 50%, then they're so far apart, they basically assort independently, because there is always going to be a crossover between them because they're super far apart. And on average, there's at least one crossover per chromosome. Linked genes are definitely on the same chromosome, therefore, and unlinked genes are either on completely different chromosomes or are so far apart on the same chromosome, they're almost always separated by a crossover during meiosis. So let's look at how you calculate recombination frequencies. And to understand that, you have to understand what a test cross is, because that's how you make a genetic linkage map. And a test cross is basically, we've talked about this briefly earlier, but I want to talk about it a lot more now. A test cross is essentially a mating between one individual that's heterozygous for two or more genes, and a second individual who is homozygous, recessive for all of those same genes. So an example of that is shown on this slide. We have a parental generation where we have two big A's, two little B's. We're crossing that to something that is two little A's, two big B's, which gives us an F1 progeny of big A, big B, little b. That's a dihybrid animal. Now, a dihybrid animal, or organism, I guess, is heterozygous for two genes. If we cross that individual to a homozygous recessive individual, that is a test cross. So that's the name for this particular cross. We're only dealing with two-factor crosses, as I mentioned earlier in this course, so that's all you need to worry about. So a test cross is basically a dihybrid organism crossed to a homozygous recessive organism. Sorry, I'm eating cheese. So if two genes are not linked, let's look at what happens during a test cross. So same example I just showed you. We're going to take these two parents, we're going to make them together to give us this F1, a dihybrid animal. If I asked you what gametes this guy could make in an unlinked situation, you would give me all the combinations, right? Big A, big B, big A, little b, etc., etc. And you would tell me that independent assortment tells you the gametes have to be present in equal amounts, 25% each. Now, if I was to ask you what gametes the homozygous recessive organism could make, you would say there's only one kind, little a, little b, and that's it. 100% of the gametes are going to be that. Now, if I was then to do this test cross and I was to write out a Punnett square for it, I should come up with something that looks like this. So in this cross scheme, the F2 generation would be all of these gametes crossed to this single type of gamete, giving us this relatively small Punnett square, and you would get four differently looking individuals, and the ratio would be 1 to 1 to 1 to 1, because it's a test cross. So 25% of them would look like this, whatever the genes A and B mean in terms of phenotypes. So that's an unlinked set of genes. Let's do the same thing with a pair of linked genes. In order to illustrate what's going to happen here, we're going to do it in the context of a question. So here's the question. Two genes are located on the same chromosome, same linkage group, in other words, and they're known to be 12 map units apart. Okay. And the question is then this. A homozygous dominant individual was crossed to a homozygous recessive individual to produce dihybrid offspring, which you then do a test cross with. And then if this final cross produces 100 offspring, how many would have the genotype big A, little a, two little b's? Bearing in mind that these genes are 12 map units apart. So here's how you answer this question. We're going to draw out a cross scheme similar to ones that you've seen before with one slight difference. So here we have our one homozygous parent, right? Homozygous dominant. If I was to write out this genotype and look at the chromosome, then I could write it out like this, right? Here's the chromosome, got one from mom, one from dad, but we're homozygous big A at this locus, and we're homozygous big B at the other locus. If we look at the other individual, homozygous recessive, we can do the same thing, draw out the chromosomes. It's homozygous, so it's pretty easy. Both chromosomes look like this. And then if I was to cross these guys together, right, I would get big A, big B, little b. You guys know that. And that's because a big A and a big B comes from this parent, and a little a and a little b comes from this parent. But if I was to write down the gametes now, well, actually, let's just do the test cross. So then we're going to take this guy and we're going to do a test cross like it says in the question. And so the first thing that I have to do is figure out what gametes could be made in each case. So in the case of the dihybrid individual, I'm going to draw out the chromosomes and we're going to look at what's on each chromosome. I get obviously one chromosome that looks just like the parent, big A, big B, chromosome that came from the other parent, little a, little b. Now, if a crossover takes place, it's going to leave us with these two possibilities because we're going to swap two pieces of DNA around, right? So now we have big A, little b, little a, big B. Now, if I look at those in the context of parental versus non-parental chromosomes, this is what I see. The top two are non-recombinant. They look exactly like the parents up here. And the little a, little b up here. And the recombinant ones are down here. These are the non-parental ones that it can only arise by a crossover taking place. Over here, of course, there's only one possibility for this other organism in the test cross, which is exactly the same as the one up here. There's just a little a, little b. Nothing else is possible. Now, what you're told in the question is that A and B are 12 map units apart. What does this mean? This means that they have a recombination frequency of 12, which means 12% of the time you're going to see a crossover occur. Now, think about what that means in terms of these gametes. 12% of the time you're going to get a recombination event because they're 12 map units apart, which means when I look at these four different types of gametes, I see that 6% of them have to be big A, little b, and 6% of them have to be little a, big B. Add those two 6% together, you get 12. 12% of the time there was a crossover, and it must be split equally between these guys because they literally exchange DNA with each other. That means that 88% of the time, there won't be a crossover. Split that between the other two chromosomes, and you get the idea that 44% of the gametes would look like this, 44 would look like this, 6 and 6% would look like those two. That adds up to 100% of all the gametes. It's much easier to figure out what's going on in the other half of this test cross because 100% of the gametes are going to look like that, right? So if I was to take this to the F2 generation like we did previously, now the Punnett square looks the same, but the numbers are going to be different. And I color-coded this so you can see which alleles came from where, right? But what you see is that you get the same 4 classes of phenotypes, but because these genes are now linked, you don't get a 1-to-1-to-1-to-1 ratio because things are not mixing equally. 12% of the time, you'll see the recombinant types, and so they're shown at the bottom here. To go back to the question you were asked, if this final cross produces 100 offspring, how many would have the genotype big A, 2 little b's? That would be 6%, as shown right here. So you can play this through a couple of times just to make sure you understand what's going on. Hopefully that makes some sense to you. I'm going to go through another example of something slightly different in just a second. So because we're now starting to understand the difference between classic Mendelian inheritance and inheritance that takes linkage into account, you can start to see how you can use statistical tests like the chi-squared test to figure out if genes are linked or not. And you can use that because remember what the chi-squared test does. The chi-squared test is going to tell you if your observed values and your expected values are the same. In other words, does your prediction match what you see in your experiment? So if you want to see if 2 genes are linked, you basically take the initial viewpoint that classic Mendelian inheritance would hold. And you're going to see if your numbers are different from that or not. So in a test cross, as we mentioned earlier, classic Mendelian inheritance would suggest a ratio phenotypically of 1-to-1-to-1-to-1 of the offspring. So if you had 400 offspring, you'd expect to see 100 in each of those phenotypic classes. So we're going to start with that expectation in the next problem. We're going to generate a p-value, and if that p-value is very, very low, if it's below 0.05, then we would expect linkage to be occurring, because a p-value of less than 0.05 suggests the two sets of numbers, the observed versus expected values, are significantly different. So let's go through a simple example of that kind of problem. In a test cross, in which a dihybrid individual is crossed to a homozygote recessive individual, they got the following results for the different types of progeny. Gives you the actual numbers of progeny down here. Total number of progeny in this experiment is 7,527. And the question here is, it's telling you to use the chi-squared test. Oh, thank you. Oh, I'm celebrating the end of the lecture with a lovely glass of wine. So the results are as follows: 7,500 progeny. We're going to use the chi-squared test to figure out if these genes are linked or not. And if they are linked, we're going to calculate what map distance they are apart. Here's how you do it. if the genes are unlinked, so if they're working by Mendelian rules, we would expect a 1-to-1-to-1-to-1 ratio in the test cross. That was my glass of wine. So what does that mean from a number perspective? If we took, if that was true, we would expect to see 7,527 divided by 4, which is 1881.75 in each category, which we clearly don't see, right? It's not even close. So we don't expect these to be sorting independently. But we'll plug it into the chi-squared equation anyway. So we have observed minus expected squared divided by expected. And we're going to do that for all 4 different classes of offspring. Going to add them all together to give us that chi-squared value. Crunch those numbers, it comes to 1,707.4. Of course, then we've got to plug that number into the probability table. We did that in the previous part of this class. This time, we've got an enormous chi-squared value. The degrees of freedom in this case is going to be 3 because it's the number of phenotypic categories minus 1. And where does the number 1,700 fit in here? Well, you can see the numbers are going up, right? But the null hypothesis becomes rejected when the chi-squared value gets above 7.8. So we are way below that, which means our p-value, if you look at the p-values at the top here, is going to be very, very small because it's miles over this side. What does that mean? That means that the observed versus expected numbers were significantly different, which supports linkage because the null hypothesis is rejected. In other words, there was a difference in the expected versus observed numbers. So the question actually asked you to use the chi-square test to predict if these genes are linked. They are. And if they are, what map distance are they apart? In order to calculate that, we go back to the equation we looked at before, which is map distance or, hold on one second. I just realized that, in fact, I missed out something down here. Let's just repair this slide. Okay, to calculate the map distance or recombination frequency, number of recombinant offspring divided by total times 100. I told you earlier that recombinant offspring are always lower in number than non-recombinant offspring. That makes sense because recombination is a very relatively rare event. So in order to calculate this, I look at the 2 lowest categories of offspring because I know these have to be the recombinant ones because their numbers are lower. Add those together and divide by the total offspring, and you get 26.2 map units or 26.2 centimorgans apart. So that part of the math is pretty easy. So that's how you answer questions on linkage and mapping. So there is an assignment this week, which you guys are going to do by yourselves. Good luck. And yeah, I'll see you next time.